Hey everybody, thank you again for joining me today. I am Pre-Med Mama, and we are still in the Kaplan Biology Review Chapter 1 section. We are in section 1-2 talking about eukaryotic membrane-bound organelles, and I am picking up today with Endoplasmic Reticulum, the Golgi Apparatus, and Peroxisomes. All right. So jumping right into the endoplasmic reticulum. This is also called the ER for short, not to be confused with the emergency room. But the endoplasmic reticulum is in fact a kind of convoluted, busy place. So the ER is a series of interconnected membranes that are actually contiguous with the nuclear envelope. The double membrane of the endoplasmic reticulum is folded into numerous invaginations, creating complex structures with a central lumen. Now there are two varieties of ER. There's a smooth and a rough. The rough ER, also called RER, is studded with ribosomes. These permit the translation of proteins that are destined for secretion directly into its lumen. On the other hand, the smooth ER, or SER, lacks ribosomes and is utilized primarily for lipid synthesis, such as the phospholipids we find in the cell membrane. And it's also important for detoxification of certain drugs and poisons. Finally, the SER, or smooth endoplasmic reticulum, also transports proteins from the RER to the Golgi apparatus. All right, next, the Golgi apparatus. The Golgi apparatus consists of stacked membrane-bound sacs. I like to think of the Golgi apparatus kind of like an Amazon warehouse, what I imagine Amazon.com warehouses to be like, because these organelles are kind of like the place where their packages are received, packages are opened and repackaged, products are repackaged, and then those packages are shipped out once again. So let's get into that a little bit more in detail. Materials from the endoplasmic reticulum are transferred to the Golgi apparatus in vesicles. Once they're in the Golgi apparatus, these cellular products may be modified by the addition of various groups, including carbohydrates, phosphates, and sulfates. The Golgi apparatus may also modify cellular products through the introduction of signal sequences, which direct the delivery of the product to a specific cellular location. That's right, guys, signal sequences are kind of like specific addresses, right? So again, if we're using that Amazon analogy, a signal sequence would let a vesicle know that it's destined for this person's house instead of that person's house, or this destination or location versus another one. After modification and sorting in the Golgi apparatus, cellular products are repackaged in vesicles, which are subsequently transferred to the correct cellular location. If the product is destined for secretion, then the secretory vesicle merges with the cell membrane, and its contents are released from the cell via a process called exocytosis. You might want to look at an image either in your Kaplan book or online, if you can find it there, to see what the relationship between lysosomes, the endoplasmic reticulum, the nucleus, and the Golgi apparatus look like. A key concept that the book wants you to keep in mind is that not all cells have the same relative distribution of organelles. Form follows function. For example, cells that require a lot of energy for locomotion, such as sperm cells, may have high concentrations of mitochondria. But cells involved in secretion, such as your pancreatic islet cells and other endocrine tissues, have high concentrations of rough endoplasmic reticulum, or RER, and Golgi apparatus. Other cells, such as red blood cells, which primarily serve a transport function, have no organelles at all. And finally, we're going to talk about peroxisomes. Peroxisomes, guys, contain hydrogen peroxide. One of the primary functions of peroxisomes is the breakdown of very long-chain fatty acids via beta oxidation. Peroxisomes participate in the synthesis of phospholipids and contain some of the enzymes that are involved in the pentose phosphate pathway, which we'll discuss in Chapter 9 of MCAT Biochemistry Review.